Hello, hi everybody. Good morning, good afternoon, or good night, depending on where you are now in the world. Um, I will just share my screen to give a brief presentation of the Pharmacological Education Initiative. Can you see my screen? Okay, so hi everyone. It's a great pleasure to have all of you here. My name is Deidiane Elisa Ribeiro. I am the coordinator of the Pharmacological Education Initiative, and I would like to briefly present this project. The Pharmacology Education Initiative is a new educational program launched by the Pharmacological Research family of journals from Elsevier in collaboration with the International Union of Basic and Clinical Pharmacology. The main objective of our program is to provide and disseminate the free access to high-quality content on pharmacology for students and research worldwide. To achieve this goal, we are promoting a series of webinars on methodology and basic and clinical pharmacology research over this year. So, of course, that nothing of this would be possible without the amazing team working on the Pharmacological Education Initiative, along with the full support of the editorial board of the Pharmacological Research family of journals, particularly Emilio and Anne-Marie, who have guided us through every step, and also our invaluable collaborators, Elsevier and IUPHAR. So, for more information and updates, please join us on our social media. And without further ado, I'm honored to declare the 2025 webinar series of the Pharmacology Education Initiative open and invite Professor Emilio Clement to speak now.
Thank you very much, Deji, for this organizing this wonderful initiative. I bring the greetings from the International Union of Basic and Clinical Pharmacology, and I'm particularly proud of this series of webinars as they are part of an ongoing work by International Union of Pharmacology to develop programs of education. And this one is particularly addressed to investigators who are in their prime, that are starting their career, to give them ideas and ways on how to develop their own research programs. And of course, it's open also to everyone with a keen interest in pharmacology. And I'm particularly happy that we were able to set up a collaboration with the biggest ever publisher, which is Elsevier, that shares with IUPHAR the same keen interest in developing education in pharmacology. And my special thanks for Deji, who is organizing the whole series, and of course to Ahmed, the first speaker of this series that will speak today. And without any further ado, I pass the microphone to Anine to introduce the speaker.
Hi, and thank you for the introduction. Today is an amazing day where we kick off a series of webinars in pharmacology. And today is so special. The guest is so special to me. He was my PhD advisor. I've known him since 2018. And since then, we've been working together. And since the first time I've heard him talking on the stage, I knew that I want to learn from this guy and I want to work with him and I want to be mentored by him. So it's a great pleasure to introduce Professor Ahmed El-Yazbi, Professor of Pharmacology and Therapeutics at Alamein International University in Egypt and Adjunct Professor of Chemistry and Biochemistry at Texas Tech University. If I want to talk about Dr. Yazbi, it's gonna take hours and hours to not consume time, and let him give us his great talk. Please, Dr. Yazbi, the floor is yours. And thank you for being with us.
Well, thank you very much, Haneen, for the generous introduction, and many thanks to IUPHAR and Pharmacological Research and Elsevier for this invitation. I'm indeed very happy to be here with you today, and I will start right away with sharing my screen and moving into slide mode. So everybody can see my screen now. All right, so
No,
let's get going.
Not yet, I guess.
Okay.
We can't see the slides. Your slides.
You can?
No.
No? Is it a problem everywhere? Emilio, can you see the slides?
I can
No,
see the slides.
they disappeared.
Disappeared?
Actually,
They disappeared. Okay.
Yeah.
Hang on. Let me see what the problem is.
now okay. Now okay, you can start. I think they are there.
Yeah.
But this
Yes.
is I think they disappear when we go into the presentation mode. Let me do this and let's see what happens.
They are there.
Okay, hang on. How about now?
Yeah, maybe if you click to the next slide.
No,
we
hang
can-
on. I'll fix it. Hang on.
Okay.
Yep,
They
hang on.
some of the guests say that they can see the slides, so there is no problem. Please carry on.
All right. So okay. So this is again thank you to the Pharmacology Education Initiative, which is a great and very valuable initiative. I hope many people make use of this valuable resource in the coming months. As my colleagues have indicated, it's a collaborative program from Elsevier and IUPHAR. And the title today that we're going to be talking about is Exploratory Clinical Pharmacology for Drug Repurposing. We're going to be discussing feasible approaches and insights. What we are going to be doing today actually is a quick brainstorm using issues from two case studies that we've been dealing with for a while now, and these two case studies deal with research related to mild cognitive impairment, and that involves repurposing of anti-diabetic drugs for mild cognitive impairment, and for identifying toxicity mechanisms and therapeutic targets for the mild cognitive impairments resulting from environmental pollutants causing neuroinflammation. What this talk is not, it is not a comprehensive set of guidelines, so it is not going to tell you what you need to do and give you a detailed information about how to approach clinical pharmacology or how to approach drug repurposing. It is not inclusive of everything that could be or can be done. It is not guaranteed. What we've done is not guaranteed to work in all situations, and it's not guaranteed to work in environments outside our environment. However, we wanted to share our experience with you and share with you what worked for us, and hope to generate some good interaction and some ideas for discussion and for future research. So why do we go to drug repurposing? There is a global challenge to drug discovery. Like there are increased attrition rates. There is a lack of ability to correlate physicochemical properties to toxicological and clinical success. So there are a lot of publications. There's a huge publication output that has grown through the years, over the years. And many papers describe new molecules, describe new drug candidates, but very little a very little proportion of these drug candidates actually make it even to clinical trials or phase 1 clinical trials, because of the lack of ability to get good toxicological properties or to get good clinical success. So there is an increased time to bring new drugs to the market, especially, and that this is especially problematic in certain therapeutic areas. And the regulatory requirements are changing all the time, and we're going to talk about that, especially for anti-diabetic drugs in a second here. Problem now is that every dollar spent on pharmaceutical R&D returns less than $1, and that decreases and reduces the incentive for research into new drug discovery. And hence, the solution became to look at older drugs, so drugs that are already existing. And this is called drug repositioning, reprofiling, retasking, or repurposing. There are different names for the same thing. Basically, it's a strategy to identify new uses for approved drugs outside the scope of the original indication. So this drug was approved for disease A, but maybe it can be useful for disease B or C. And this strategy is advantageous because there's a lower risk of failure. You already have a drug molecule that is already been found to be sufficiently safe. We're just going to do some preclinical testing. We're going to do safety assessment in this particular disease that we're targeting. The formulation development has already been completed, so there is a lot that we're bypassing here. So there is less investment required, that may be estimated to be $300 million compared to $2 to $3 billion for a new chemical entity. Moreover, when you work on repurposed drugs, you might end up finding new targets. And we're going to talk about something like that today. And this new target that has not been apparent may trigger thinking about new drugs to exploit this new target or this new pathway. Typically, in the old days, drug repurposing happened by coincidence. So you have a drug like zidovudine, for example, that was originally developed for cancer, but then through in vitro screening of compound libraries, we found out that it can work for AIDS, for HIV. It can act as an antiretroviral, inhibits the viral DNA polymerase, and so on and so forth. Happened with many drugs like the minoxidil developed for hypertension, but found its use in hair loss. Sildenafil developed for angina, but then worked for erectile dysfunction. Thalidomide, which was developed for morning sickness and was associated with the terrible side effects of the congenital malformations, but then made a reemergence in cancer, for instance, multiple myeloma. Celecoxib developed for inflammation, but then made another appearance in familial adenomatous polyposis, and so on. But this happened serendipitously, so it was every now and then, not structured. Now there are many structured approaches for the identification of candidate pre-existing molecules for a given indication. So you can mechanistically assess them in preclinical models and then go straight to evaluation of efficacy in phase 2 clinical trials. And there's a lot that could be done. So you can do experimental approaches, maybe through binding assays to identify relevant drug targets, through phenotypic screening in disease models or like animal models or even cellular models. But then there is a lot of computational approaches as well that you can that can be done through pathway mapping, network analyses, genetic associations, signature matching through the effect of the drug on transcriptomic, structural, or adverse effect profile, and matching that to the disease phenotype, and so on and so forth. But we as pharmacologists, we always like to deal with a solid mechanism of action, right? And we want to do experiments. We want solid experiments. We want solid results. We want to explain things. So there are barriers. There are always barriers against drug repurposing and against experimenting with drug repurposing, specifically and especially when that experiment or that investigation involve human subjects. So there might be certain drug characteristics that would prevent you from carrying this investigation in the way that you would hope to do. So side effects, for example, might deter the use of the drug. We've done quite a bit of work on anticoagulants, for instance, on direct oral anticoagulants, and there's always the barrier of the bleeding side effects that would prevent or would form a significant barrier in using them for as anti-inflammatories, for instance. There are regulatory issues in certain countries, certain settings, preventing you from using the drug out of the scope of its initial indication. Sometimes there might be a lack of mechanistic extrapolation from preclinical models, just by virtue of differences in the genetic and protein makeup between animal models and humans. There might be certain targets that are not present in rodent models, for instance, or other mammalian models, and I will be talking about an example of that today as well. Problem is, you might find an effect that is interesting, but you don't know what's the target, what is mediating that effect, and what are the other outcomes that you need to measure. And this is a big challenge, because if you don't know which target and which outcome to measure, then you're faced by significant barriers in sample size calculation, for instance, and for mechanistic interpretation of the effects that you are ending up with. There are always ethical challenges precluding structured patient exposure that would prevent investigation in this direction. So what we are going to focus on today, as I said, is the mild cognitive impairment that is happening in the context of diabetes and metabolic disease. And we are involved in extensive research. We have been involved in extensive research in that field for quite a while now, just because looking at this map here that I borrowed from the International Diabetes Federation Atlas, you will see that our region here, the Middle East and North Africa, and in Africa, both regions remain among the highest regions in the world, with an estimated increase in diabetic patients, where areas where, like in the United States, for instance, that had higher rates of obesity and metabolic challenge, things appear to have abated over the years, so they have gotten over the worst of it, but we here are still seeing the highest rates of surge of metabolic disease. And this is associated with a huge expenditure, huge growth in projected expenditure in the next 20 years. And our economies are already challenged, so we need to find solutions. And the problem with metabolic disease and diabetes specifically are the complications. And our outlook is not very well. It's pretty gloomy, because even with the tightest glycemic controls, trials and studies tell us that your diabetic cardiovascular complications would still progress. And there is a significant proportion of patients that already present with microvascular complication at initial diagnosis, patients who even are not diabetic but might show some impaired glucose tolerance are at an elevated cardiovascular risk already. And all of these findings that have been known for a while tell us that there is an inflammatory process that commences well before hyperglycemia becomes evident, becomes manifest and overt. So we need to look at the root cause of these diseases, root cause of these complications, and address them. So complications can exist even in a seemingly healthy individual. And this view actually has been confirmed over the years with findings of large clinical studies. And the story behind these findings actually go back to 2008 with concerns with the cardiovascular safety of rosiglitazone that led to its withdrawal from the markets and triggered the FDA to issue guidance regarding cardiovascular safety of anti-diabetic drugs. So now all anti-diabetic drugs coming to the market have to do a cardiovascular safety assessment study and demonstrate cardiovascular safety. So early cardiovascular outcome trials, for instance, that were done for DPP-4 inhibitors were sufficiently powered to show safety. But I think those who are involved in these trials later on thought that let's increase the power and detect if there is a cardiovascular advantage to some of these drugs. And indeed, it was shown for GLP antagonist liraglutide, for instance, and for the early SGLT2 inhibitors, the dapagliflozin, canagliflozin, and empagliflozin. I think it was empagliflozin that was the first SGLT2 inhibitor to demonstrate superior cardiovascular protection in type 2 diabetic patients. And these trials did not even stop at type 2 diabetic patients, since the protective effect was shown to happen independent of the glycemic effect of these drugs. So these drugs were protective, even if there is no measurable decrease in glycosylated hemoglobin. So clinical investigation was extended to include non-diabetic patients. And even in non-diabetic patients, these drugs showed equal cardiac and renal benefits. So far, the focus has been cardiac and renal complications. We started thinking about the mild cognitive impairment as a potential complication of type 2 diabetes and of metabolic dysfunction as well, especially again because of how high the prevalence of these diseases is currently in our country, and how it is projected to grow in the coming 20 years. And since no one typically thinks of diseases of the elderly, since our population is typically viewed as a young population, but we're getting older. There was a 2.56% increase in old age population since 2006, and it's projected to be about 11% by 2031. So in 6 years, we're going to see an 11% growth in elderly individuals in Egypt. And this is further compounded by the fact that mild cognitive impairment is more prevalent here. So we are looking at 35 to 50% of older Egyptians compared to the global average of around 20%. So we do have a clear and present challenge here of mild cognitive impairment. And this is occurring despite the definition of old in Egypt starts at a younger age. So our retirement age here is 60 compared to 65 in most other nations. So what's happening here? Risk factors specific to our environment include metabolic dysfunction, and we've discussed that already. It's higher than other places, and also exposure to environmental pollutants. And one of the main environmental pollutants that we see here as a challenge is the exposure to organophosphate insecticides. We know that there has been a significant increase in the use of organophosphate pesticides in our agriculture activities, and with the shifting climate patterns that we see here and varying pest infestation patterns, they are being used, or I would say abused, even more. And that would lead to neuroinflammation, and no one knows what's happening, how it is happening, or how to even approach it therapeutically. So what is mild cognitive impairment? So mild cognitive impairment are signs of, cognitive dysfunction that people typically dismiss or tend to deal with as normal, as a normal part of growing old, but it is not normal, and it signifies a higher or an increased risk of dementia and Alzheimer's disease. And if you look at the symptoms, you feel that they are things that you've seen in people, and heck, maybe ourselves, like maybe myself, right? Forgetfulness of recent events, difficulty in finding words or names, trouble in decision making or problem solving, reduced ability to plan or organize tasks. And I know graduate students who blame MCI, blame mild cognitive impairment all the time, but they're too young for that, I guess. Frequently misplacing items, increased confusion in familiar environments, and challenges in following a conversation or a storyline. So these are symptoms that we see quite a bit. So what can we do? How can we use? The first question is, if anti-diabetic drugs, like newer anti-diabetic drugs, were successful in mitigating cardiovascular and renal risk, cardiovascular and renal events, would they also be successful in addressing mild cognitive impairment? Are we able to demonstrate a clear mechanism through which these drugs can address mild cognitive impairment? So that was our first question. That was our first preoccupation. And although we've shown that in animal models, we were hoping to be able to show it in human subjects as well. And so that led us to the first case study that we're going to be talking about today. And that was the easier case study. So I brought it first. So we're talking about drugs that are approved for diabetes, and we're talking about a complication that is present in diabetic patients. So that's easy enough. You're going to have patients who are potentially taking these drugs, who are also potentially going to develop these complications. So we do have the patients. But what is it that we're going to look at? So we're going to look at their cognitive function. Do we have a measure for the cognitive function? Again, this is the easier question we do. We do have the Montreal Cognitive Assessment Battery, the Montreal Cognitive Assessment Score that measures the cognitive ability, and that gives you an objective measure of their cognitive performance. So all we have to do now is to find a group of patients who are taking the target drugs that we're looking at, and we were mainly interested in SGLT2 inhibitors. We were hoping to be able to study GLP-1 agonists as well, but because of economic and environmental constraints, we were not able to find enough patients in Egypt who are on GLP-1 agonists because they are very expensive. So we went to the next best thing, which are the DPP-4 inhibitors. Anyways, the idea now is to find a group of patients who are on these drugs and a group of patients who are not taking these drugs and study evaluate their cognitive ability and determine if there is any significant difference. Now, that begs a few questions. What is the sample size that we're aiming at, and is it feasible or not? And what is significant? What is significantly different? Are we just looking for statistical significance or clinical significance? So that's a typical question that faces you whenever you're looking at an outcome in human subjects. Like on the bench, we're very happy with statistical significance in animals or cell experiments or binding studies. We're very happy with the statistically significant difference. But now we have to remember that we're dealing with a human subject who is probably going to be paying a price for getting on that therapy, and that price is not necessarily only financial, probably also a price and side effects, potential interactions, and so on and so forth. So what is clinically significant? Fortunately, again, because this is the easy case study, there has been clear definitions of a clinically significant difference in MoCA scores, and that is 2 points. So if you have a 2-point shift in a MoCA score, then that means that the patient is performing much better clinically speaking. So we used that cutoff, and we ran our sample size calculation, and we ended up with numbers that are sufficient to detect a 2-point change in the MoCA score. And we were lucky to end up with patients who are receiving metformin monotherapy and patients who are receiving metformin with either DPP-4 inhibitors or SGLT2 inhibitors. And we also had non-diabetic subjects that we've used as a background control here. So we were hoping to demonstrate a difference between these two groups and then look further into the potential mechanism of action of these drugs and how they induce that difference, if it existed. So these were our patients, how they are distributed between metformin, between the DPP-4 inhibitors here, and between the SGLT2 inhibitors. And as you can see, our patients were distributed among different molecules and different strengths, and reflecting the real-world nature of the study. And that's another challenge as well, because, it's going, unless we have like strong financial support, and unless we have ethical approval, we won't be able to get patients on certain strengths. But again, we were looking at a real world situation, hoping to uncover a resilient effect of these drugs. So what did we find? We found that type 2 diabetic patients on metformin monotherapy did perform worse cognitively compared to the healthy volunteers. And that difference here is actually more than 2 points, right? So they had a clinically meaningful reduction in their cognitive ability, while those on the combination therapy that included the newer anti-diabetic had a better performance. So we had a basis for our study now, and this is at baseline, right? And this difference here, you might argue that those who are on combination therapy might have better glycemic control compared to their counterparts on metformin monotherapy, but that was not really the case here, because despite being on combination therapy, their random blood glucose level, their glycosylated hemoglobin was still high, comparable to those on metformin monotherapy. So the actual care for the glycemic control, you might say, was not compromised for those on monotherapy. So they were still receiving the typical type 2 diabetes care, aiming for their blood glucose levels. Granted, it was not working very well because their averages are above 8, but still it was not different among the two groups. We looked at the typical suspects here. So inflammation, C-reactive protein, and IL-6, and we focused on C-reactive protein and IL-6 because they are linked in the literature to reduction in cognitive function in type 2 diabetes patients, but then they were not really different among groups. We looked at complications, so we looked at liver function, we looked at the kidney function, and there didn't really seem to be a difference among these patients. And that means that our patients, either the ones on metformin monotherapy or the ones on combined therapy, are not far along in the pathogenesis of their type 2 diabetes. And that means that they are still, they do not still show overt complications. And since they do not show overt complications, these drugs are improving their cognition by mechanisms other than attenuating the typical complications that you see. So we're trying to rule out common explanations here, or actually trying to see what's going on, but common explanations did not work for us. So is the effect resilient? Yes, we do think it is resilient because it is happening with molecules that are representative of the same class, but we're not using the same molecule, different molecules. It is resilient because it's happening with different strengths that are used for these patients. Are there confounders? Of course, there are confounders, and we're going to show you that we corrected for them. And the other question is, how fast is that effect? So in terms of the duration or how fast the effect is, we tried to look at whether the MoCA score would vary with the duration of therapy. So for those on metformin or those on combination therapy, we didn't really find any correlation among the duration of therapy of metformin or the combination therapy and the MoCA scores. So it told us that probably the effect that we see on cognitive function is going to be rather fast. There's not been we followed those patients up, like we followed up on the metformin monotherapy patients and the combination therapy patients for 6 months to see if their cognitive function would vary. And we didn't see it really, like between baseline and follow-up, there was no variation in cognitive function in the different groups. And still the significance was maintained. So monotherapy group was performing worse than controls, but not the ones on the combination therapy. So that means that newer anti-diabetic drugs, the SGLT2 inhibitors and the DPP4 inhibitors, indeed did have an effect on the MoCA scores or the cognitive performance. But then when we looked at the glycemic control, we've seen an overall decline. So the worse your glycemic control is, the worse is your cognitive improvement probably. But then this did not make a difference in our findings because both groups of patients still had bad glycemic control. One more thing that we found is also that the MoCA scores or the cognitive performance decreased with age. And that is to be expected, of course. But interestingly, the group of patients on newer anti-diabetic drugs were older. So even though they were older, expected to have lower cognitive performance, they still had better performance just by virtue of being on these drugs. We looked at the blood lipid levels, the total serum cholesterol, and the LDL cholesterol. And it was lower in the patients treated with the newer drugs. And then the question arose whether these factors could have contributed to the effect that we've seen. And when we correct for blood lipids, we don't see any effect for blood lipids affecting the MoCA scores. The other question was, these patients might be taking statins. So part of the guidelines for treatment of type 2 diabetes involves statin therapy. So we've corrected for statin therapy as well. And we showed that statin therapy didn't really affect the MoCA scores within the different groups, but the general differences remained. These patients that are only receiving metformin performed worse, and those who are receiving the newer anti-diabetics perform better. So we were basically at a loss now. Where do we go? What do we do? All the common explanations did not help us. We don't know how these drugs are producing their effect. So there's in the literature, you will find some studies talking about an anti-inflammatory effect, although to the best of our knowledge, they have been inconclusive. Like if you look at meta-analyses, looking at the effect of SGLT2 inhibitors, for instance, on serum cytokine levels, they say that no, there is no effect. There's no net effect on inflammatory cytokines. So as pharmacologists, although we are happy that these drugs are improving the cognitive function, we cannot be satisfied by that without knowing how they actually work. So we've resorted to, we've been lucky to have collaborators that help us resort to an omics or a systems approach. So we were able to run metabolomics, lipidomics, and proteomics and look at the changes in the patient serum. And what we found basically when we ran metabolomic studies, we found that in type 2 diabetic patients who only receive metformin, the purine metabolism, the purine catabolic pathway is enhanced, and that led to an increased amount of xanthine that we were able to measure to the point that xanthine serum level was actually diagnostic of type 2 diabetic patients receiving metformin monotherapy. On the other hand, one of the main things that popped out when we compared those patients on metformin monotherapy compared to those receiving the newer medications was that the glutathione metabolism actually, again, was enhanced in the newer antidiabetic group compared to the metformin monotherapy. And one marker that jumped out was pidolic acid, or also known as pyroglutamic acid. And the pyroglutamic acid is a product of oxidation. So it gets oxidized as more oxidized glutathione is produced. And again, the pidolic acid is diagnostic of the monotherapy group here. So that basically indicates that the patients on the type 2 diabetic patients in general, they have a higher oxidative stress, and that oxidative stress was attenuated and mitigated in the group receiving the newer anti-diabetic drugs, probably because these newer anti-diabetic drugs, especially the SGLT2 inhibitor group, would produce a fuel shift that decreases the flux through the pentose phosphate pathway, and reducing the flux through the pentose phosphate pathway would decrease purine breakdown. And this is important because purine breakdown, especially in the brain, is associated with the production of, is actually xanthine oxidase is considered a main source of oxidative stress of reactive oxygen species in the brain, and the increased oxidative stress would contribute to neuroinflammation, neuronal apoptosis, and these kinds of mechanisms that we've consistently shown in our models of metabolic dysfunction in rodents. The question now is, can you prove it? Do you have an independent marker that would tell you that, yes, this is what is going on? And again, this has been fairly easy so far, because when we measured the total antioxidant capacity in these patients, we found that indeed total antioxidant capacity was attenuated in type 2 diabetic patients receiving metformin monotherapy, but then it improves in patients receiving the combination therapy. So just receiving SGLT2 inhibitors or DPP4 agonists, the total antioxidant capacity improves. So that was sort of an independent validation outside of our metabolomic study. And when we correlate the antioxidant capacity with MoCA scores, you find a positive correlation with that. So the better your antioxidant capacity, the better are your MoCA scores. And this is an example of, again, finding a target for intervention that that you haven't thought of before, or you haven't thought of in this particular indication before, which is the xanthine oxidase. So our findings would support the case of using xanthine oxidase inhibitors for mild cognitive impairment. And indeed, there's some research that looked into the inhibition of xanthine oxidase for treatment of Alzheimer's dementia, and our findings follow along the same line. I'm going to ask you, are these findings exciting? They are, but not so much, not to me at least, because among the audience, I could tell that maybe one or two could have sworn that these medications reduce oxidative stress. So it is fine. It's good that we've shown it, but you know what? It's not huge, to me at least. So we thought we wanted other targets. We wanted some wicked findings. So we went on further, and we looked at the proteomics, changes in the proteins, and what happens to the proteins. Do they support our findings or not? And we've done some comparisons, and we focused on the differentially expressed proteins, and we mainly focused on those proteins that were preserved in the combination therapy group. So the proteins that are preserved did not change compared to the healthy controls, and those proteins that were attenuated or changed in the monotherapy and then reversed in the combination therapy group. So we focused on that group of proteins that compared to controls did not change, or changed in the monotherapy but then were adjusted by the combination therapy. And when we studied the pathways enriched in these proteins, two pathways popped up: the complement and coagulation pathways. So to me, I would say this was very good news because of my interest in thrombo-inflammatory pathways. We've seen that in early metabolic dysfunction, in prediabetes, for instance, the adipose inflammation and the low-grade inflammatory process that happens includes a very strong thromboinflammatory component. And we have some animal experiments where we can attenuate that by using direct oral anticoagulants. I've alluded to that earlier. Now we have evidence from our experiment in human subjects that the complement and coagulation pathways are activated in in these patients, and they might indeed be contributing to what we're seeing, but we still have to prove that or show that at least. So what we've tried to do afterwards is we've tried to look at our drugs, the three drugs that are represented in our study. So the vildagliptin, the empagliflozin, and the dapagliflozin, and to see if they carry potential of interaction or interference with these pathways. So we followed a simple approach that we've developed, that we've used before. And that simple approach is to try to use the Swiss target design tool for target prediction. What are other targets for these drugs other than those targets or other than those canonical targets? So basically, this is what drug repurposing is about. We're trying to look for other targets. So we ran the algorithm, and the algorithm comes up with a list of targets with probability ratings, and we take the top probability ratings for every drug, and we start running to see which pathways are enriched in those top probability targets. So for vildagliptin, for instance, again, complement pops up. For dapagliflozin, again, you see complement pathway among the significant pathways. And for empagliflozin, again, you see the complement pathway among the significant pathways. So now the question would be, what are those targets? Which targets? What is going on? So what we've done afterwards is we went, so this is the list of proteins that were preserved or reversed in terms of expression in patients that are treated with the SGLT2 inhibitors or the DPP4 agonist. So these are the same. So here, here, and here, this is the same set of proteins. And what we've done is that we've mapped the interactions among these proteins that we actually found in the patients, that we actually detected in the patients, and map the interactions in between these proteins and those potential drug targets for the three drugs that are involved in the complement pathway, right? So we know that the complement pathway is enriched in these proteins, and we selected those targets of the drugs, we selected the potential targets of the drugs that are enriched in the complement pathway, and we mapped the interactions. We wanted to look at the interactions and see what these protein-protein interactions are looking like computationally, right? And we started developing an interest in hub proteins. And hub proteins are those proteins who develop at least twice the average number of edges here, right? So at least twice the number of interactions. And consistently in the three cases, in the three drugs, two proteins kept popping up, phycolin 3 and factor 8, coagulation factor 8. So what is phycolin 3? So phycolin 3 is basically a pattern recognition receptor. It's a lectin that is part of the lectin complement pathway, and coagulation factor 8 is a coagulation factor, right? So these two kept popping up, and they represented the proteins that are probably affected by our drugs of interest and altering the pathophysiology in our patients. So we looked again at the diagnostic value of either drug. So you see that phycolin 3 is significantly reduced in the monotherapy group, and it gets restored in the group the group of patients receiving the newer drugs. The same pattern holds for factor 8, and we run ROCs, you run receiver operator characteristic curves. Both proteins are diagnostic. So which is it? Which one? And can we validate these findings? That was a tough question. We started thinking about what to do. What can we do? What can we do with this data? How can we push it further? How can we prove that actually one of these proteins, one or both actually of these proteins is working? And that's when that was when I turned to one of my very good friends who is a very smart drug a medicinal chemist, and He developed a very simple in silico experiment where we've actually started docking these drugs against the protein, the 3 drugs against the 2 proteins, and compared the poses of these drugs to natural ligands of these proteins. So phycolin 3 basically is, again, a pattern recognition protein, a lectin, that detects a carbohydrate pattern on a microbe, for instance, and then starts to recruit the complement pathway and trigger an immune response or an inflammatory response. Phycolin 3 is particularly important because it can interact with galactose, and galactose is one of the markers that are exposed on the surface of apoptotic neurons. So if you have a neuroinflammatory state and you have an apoptotic process and you have apoptotic neurons, these neurons are probably going to express galactose, and that is going to trigger phycolin 3 and trigger that immune response. And what my friends were able to find out is that our drugs were docked much, much better than galactose. So there are glycyl moieties, right, in the dapagliflozin, vildagliptin, and empagliflozin that interact with phycolin 3 much, much better than the natural ligands. So if you have empagliflozin in the environment, in the microenvironment, they're going probably to preclude the interaction between phycolin and galactose and thus inhibit the triggering of the immune response. We could not show the same for factor 8. So the 3 drugs would interact very weakly with factor 8 and much worse than the ligands that we found available for factor 8. So we have a case where our drugs can reduce neuroinflammation by interfering with complement pathway. And this is through the glycyl moieties that would masquerade as the carbohydrate triggers of phycolin 3, and that would stop the inflammatory pathway from being triggered. But then again, we need to prove that. We need to show that this is the case. This is not serendipity. This is not just a simple change in phycolin in our patients that happened by accident, and that we're basing all of this on. And again, we were lucky to have access to a cohort of patients who were exposed to traumatic brain injury. And traumatic brain injury patients develop neuroinflammation, develop clear neuroinflammation that proceeds or increases over time. And the cohort that we've had, the cohort of patients that we've had was a longitudinal cohort where we've got controls that do not have TBI and patients who are in day 1 post-TBI, day 3 post-traumatic brain injury, and day 5 post-traumatic brain injury. So these patients are at different stages of neuroinflammation that is potentially worse over time. And what we found when we looked for phycolin 3 in these patients is that phycolin 3 decreased over time. So indeed, the decrease in phycolin 3 that we're seeing in our patient is common to other neuroinflammatory, bona fide neuroinflammatory conditions. And this took us back to the bench, and we've scoured the literature for any indication of phycolin 3 involvement in neuroinflammation in rodent models, and we couldn't find any, simply because rodents do not express phycolin 3. The evidence that we could find regarding phycolin 3 is that it is also reduced post-stroke in humans. So right now we went back to the bench. We are trying to develop and work on rat models and prove that mechanism in a structured way. And indeed, when you look at the literature, especially recent literature from last year, there has been telltale signs in the literature that SGLT2 inhibitors modulate the complement pathway or might interfere with complement activation, albeit nobody actually had a clear target for where it is acting and how this affects the downstream activity that we're seeing. So this was the first case study. As you can see, there was a series of studies that we've performed. We were lucky to have quantitative measures, we have objective measures. We were lucky to have patient cohorts to look at. We know how the drugs act. We have certain clues as to where to go. We're going to shift now to the second case study. I don't have to finish all the slides, so if you guys getting bored, if we have to stop at any time, let me know. I wouldn't mind at all. We're going to shift now to the second case study, which involves the impact of environmental toxins, basically organophosphates that are being used a lot in our environment that get into our food, get into our bodies through inhalation and through dermal exposure, and they cause massive changes in us. They affect our population health, and we don't know how they're producing that and how this is happening. We know that they induce trigger a neuroinflammatory process, but we don't know how. They are linked to cognitive decline. They are linked to motor disorders, but again, there's no clear mechanism of toxicity. They are linked to liver and kidney dysfunction upon prolonged exposure, but again, no one knows how or where or what they are doing. So we're basically going through uncharted waters. We don't know what's going on. We're trying to shed some light by finding long-term by finding markers of early exposure. So we want to diagnose these patients. We want to show that these patients are at risk of long-term consequences of organophosphate exposure, but before they actually develop these consequences. So as you can tell, this is not as easy or straightforward as the previous study. So we don't have objective measures, right? We don't have something like the MoCA score. We are hoping to catch these patients before they develop cognitive decline. We're hoping to catch these patients before they develop liver and kidney deterioration that can be measured by serum markers of kidney and liver dysfunction. But what can we do? So we tried our best, basically. And plus we don't know what we're looking for. So we don't know what the markers are. But we tried our best. So again, we went out in the field, tried to recruit patients with known exposure to organophosphates. So our best bet was agriculture workers who sprayed these pesticides regularly, and who we know for sure have been exposed to these pesticides, and then compared them to unexposed or city dwellers who are not at least equally exposed to the same amount of organophosphates. But again, now the challenge would be the sample size. We had to take an educated guess, and we estimated the sample size guided by serum levels of interleukin-6 and CRP again. But these two were again implicated in the Parkinson's disease that is consequent to organophosphate exposure. But problem here is that the literature that report values of IL-6 and CRP report them in patients who have overt Parkinson's disease. And for that reason, when we've done our sample size calculation, we went for a 1-to-2 enrollment ratio. So we increased the enrollment for patients who are exposed, hoping, and we increased the number of patients, hoping to be able to increase our chances of finding these early exposure biomarkers. And we started looking at the usual suspects, typical things to look at, so total serum cholesterol, and the first column here is the unexposed patients, the city dwellers, and the third column is the chronic low-level exposure. We're going to end up with a middle column representing acute exposure patients, so those we recruited from our poison center, but let's just disregard them for the purpose of today. So you can see that there is no change among the unexposed patients and those exposed chronically in serum cholesterol, LDL cholesterol, triglycerides might have increased a bit. There's no change in liver function. There is no change in serum creatinine, no change in IL-6. Paradoxically, C-reactive protein is reduced in those patients with chronic exposure. So we went straight to the systems approach, and we looked at their metabolomic and proteomic profiles. And indeed, their metabolomic profile implicated changes in the pathways in the nervous tissue. So we knew then that we were on the right track, and we went to for further exploration to look at what pathways are changed. And one of the pathways that came out right away was their synaptic transmission pathway, and we've had a strong indication that these patients would suffer from again, neuroinflammation resulting from excitotoxicity due to the increase of glutamic acid. And that might be because of reduced feedback suppression by their endogenous endocannabinoid pathway. And as you can see here, these patients showed a very profound decrease in arachidonic acid, which is a typical precursor of endogenous cannabinoids. And again, this was one of the situations where we ran back to the bench. We created a rat model of organophosphate exposure, and we wanted to see whether arachidonic acid supplementation would mitigate whatever behavioral and cognitive symptoms that this rat would show. And my student is in the audience who's worked on that project, and she's going to be defending her thesis very soon. Hopefully, we're going to be showing some of these results in the next ASPET meeting in April. What else? So we want biomarkers, right? So we want proteins. So again, we looked at the differential expression of proteins here, and we were interested in the changes in proteins that have a high diagnostic capacity. And we ended up with 10 proteins who have an ROC area under curve of more than 90%. So they have a more than 90% diagnostic capacity of indicating or differentiating chronically exposed patients from unexposed patients. Very good. But again, from a pharmacological or toxicological point of view, what is the mechanism of action? What is happening? And that's when we went to running pathway analysis after looking at the different levels of these proteins. But then before we go to pathway analysis, let me bring to your attention two patterns here. Like we have a pattern that is progressive. So in unexposed patients, you find this is an actin isoform that is high. It decreases slightly in acute patients and then decreases further in chronic exposure patients. The same with a dynein, which is one of the cytoskeletal proteins in neurons. It increases progressively. Glutathione peroxidase decreases progressively, and von Willebrand factor increases progressively. Versus another pattern of change where there is a change in chronic exposure patients versus unexposed patients that does not go through the same progressive pattern with acutely exposed patients. So we developed quite the interest in these proteins, and we went further to look at the pathways involved. Again, you see here the complement and coagulation cascade jumping up clearly, plus the apoptosis, which is to be expected in the neuroinflammatory pathway. So again, the complement and coagulation pathway, the thromboinflammation comes up, and that gives us insight into what targets to look at for treatment. And these are the proteins. These are the 10 proteins that we found amongst the whole set of proteins that are changing. So these 10 proteins are the ones that are showing the highest diagnostic capacity, and they all converge on ApoE here, for instance. They all converge on complement 4. They all converge on mannose binding lectin 2, which is another isoform or another analog of 5:3. So the same pattern is maintained. We have a trigger of the complement pathway that leads to neuroinflammation. Again, which one is important? Which one is can be validated? Which one counts? We're going to have to look further into that. But we can produce some evidence, at least preliminary evidence, or get some clues from our data. Again, this is just to demonstrate that the changes in all the proteins are going together. Now the question is, what triggers neuronal apoptosis here? And the simple answer would be the DNA damaging effect of the organophosphate pesticides. And we went to an in vitro experiment to demonstrate that indeed these pesticides can produce double-stranded DNA breaks fairly fast, and these double-stranded DNA breaks would trigger the activation of the p53 pathway and the activation of the neuronal apoptotic pathway that again would probably activate the mannose binding lectin pathway and the complement pathway, leading to neuroinflammation. Tried to again validate that independently by looking at the they produce double-stranded DNA breaks by generating reactive oxygen species. And what about the total antioxidant capacity in these patients? And indeed, the total antioxidant capacity is reduced. And when we look at our markers and we try to run them into support vector machines, we find that our markers combined can have a foolproof detection or foolproof early detection of organophosphate exposure, or at least a foolproof early detection of triggering the pathways that lead to neuronal inflammation in these patients. I know that by now you probably have lost connection with me, but let me tell you one more thing that we're working on. One problem that you might argue is happening with these sort of omics studies, that you always have to rely on extrinsic databases. Like, we look at changes in proteins in our patients and we try to compare these changes to what have been found in others, like in other diseases, compare them to that databases that have been generated from other people's work. This practice in itself might bring in bias. Like, we're not doing a solid, let me not say solid. We're not doing an experiment that has a control for every step. And we might be bringing a lot of bias from these extrinsic databases. And these extrinsic databases might favor one pathway over the other, might favor one protein over the other, just by virtue of how many studies were done and what was selected, and so on and so forth. So we've been thinking about an approach to select the biomarkers independent of any influence from extrinsic databases. And that's when we came up with a way to classify our patients based on their profile. And that's when we decided to integrate a machine learning approach. So we tried to use the superior classification power of machine learning algorithms, basically convolutional neural networks, to obtain a classification of our patients from our unexposed individuals. And that's when we've rendered our LC-MS-MS chromatograms, basically the LC-MS-MS chromatogram that you end up with in a proteomic experiment. It has peaks that represent different proteins, and these peaks will have a certain retention time, they'll have a certain intensity, and then they'll have a mass spec pattern that will give you an M/Z, which is the molecular mass. So you have three values representing every peptide that you end up with in the serum of those patients. So basically, you end up with a 3D matrix for every patient, and this 3D matrix is huge, right? So it's something like that, but imagine it's formed of over 500,000 rows. And what we've done is we take these matrices, we convert them into an image or a heat map, and then we run that heat map through our algorithm, and our algorithm would classify them, telling you, yes, this is an unexposed patient, and this is a chronic patient. Then this classification is about 100% effective. The next step would be to ask the machine how it found the difference, and then pinpoint these markers as potential markers. So I'm going to conclude now because I've taken a lot of your time. What I've tried to show you in this presentation here is that sometimes you need to take unconventional approximations. It might be necessary to do that, to overcome sampling obstacles, interpretation obstacles. You always need to think about how you're going to validate your results, how these results are going to make sense. Never give up on bench experiments. We always have to go back to these bench experiments, and always take risks, right? You talk to people, take risks. Taking risks is always useful. I want to thank my trainees over the years. Many of them are in the audience. Some of them could not make it. Thank you very much for all your help over the years, and thank you to all collaborators and funders. And this is your code to download your certificate of attendance.
So thank you so much for the amazing presentation, Professor. I would like now to open for questions. If any of the anyone has, yes, Juan Essen, please. Can you talk? Someone has raised the hand.
Deja, they can only type in the Q&A box or the chat.
Okay, so please type on the chat or on the QA session, then I can read.
Ashraf, can you please write your question in the chat?
We wait him. Samia has a question. Please, Samia.
Yes, just while the others are posting. First of all, thank you. It was extremely interesting to explore this molecular landscape. I was especially fascinated into the first study, but also on what you mentioned in the second one. I work with endocannabinoids, so I was surprised with this link that you were proposing in astrocytes, and whether you were able to explore that further in some way, in addition to all the other things you have shown.
Right. Thank you very much for the question. So basically, we have developed that model there that I talked about with the dermal exposure, low-level dermal exposure of organophosphates, and we see very clear signs of astrocyte activation. And we see also a parallel decrease in arachidonic acid concentration in their serum. So our working hypothesis is that the presence of organophosphates, they do have a side effect of blocking the enzymes that are involved in the endocannabinoid metabolism, be it the fatty acid amide hydrolase or the monoacylglycerol lipase. And just the presence of the organophosphate produces an imbalance in the endogenous endocannabinoid concentration. And when you supplement arachidonic acid, you are again interfering with this imbalance, and it does have a clear anti-inflammatory effect, which reflects on their behavioral and cognitive function as well. Not only cognitive, but also motor function.
Thank you.
You're welcome.
We have one more question from Abdemi, which is in the organizing committee of the Pharmacology Education Initiative. His is, thank you for the brilliant presentation. Can you please give me more insights into the algorithm for identify candidate agents for repurposing within the context of your work?
If I understood the question correctly, the question asks about what algorithm we use to select candidates for repurposing. We have not used an algorithm to select candidates for repurposing. What we've done is we've looked into drugs with known activities, and our hypothesis was that these activities could alter the changes that we're looking at. But if you're interested in a certain algorithm, there has been a paper that was published in Cell Metabolism by one of my good friends that actually that developed a string algorithm that ran a huge drug library for an enzyme to select candidates. So basically, that study selected pimozide for increasing the skeletal muscle metabolism in type 2 diabetes. I can share the link with you later. If you were asking about the target prediction, we've used the Swiss target prediction tool, which is a free tool, Swiss Drug Design, which is a free resource that is available online that anyone can use.
Okay,
Let me see. Anyone else? I think that
there is a question by Ashraf, who managed to do. I don't know whether you see it. Otherwise, I can read it.
It's in the chat.
yes,
I can see it.
I just saw it. So Asha is asking, which stage of drug discovery, hit, lead, then clinical trials are going to be shortened or skipped if we adopted the drug repurposing concept? We are going to go directly for the clinical part, skipping the preclinical phase.
Yes, you are going to at least skip a major chunk of the preclinical phase and phase 1 trials. You're going to go straight to phase 2 trials, where you're going to try the drug in efficacy on the condition of interest, because you've established safety already, you've established dosing levels. You might need some preclinical investigation as a proof of concept just to prove that this particular drug is producing the effect that you're repurposing it for.
Perfect. Thank you so much. Anyone else have questions? So I think that's it for now. I would like to thank you so much for the amazing presentation, Professor. And I would like also to thank you all the audience that we have today, and also the organizing committee, editorial board of the Pharmacological Research Family of Journals, Elsevier, and IUPHAR. Thank you so much.
Thank you for having me. Thanks for the invitation.